Hopefully you won't have students forced to stay home in the next few years, but just in case, we're going to provide you with some neuroscience-related experiments that can be done using just household items. And of course, they can be done in the classroom as well. One low equipment option, which just takes a ruler and a partner, is to measure reaction time in response to auditory, visual, and tactile stimuli. The partner is going to hold a ruler just above the subject's hand, so pretend my two hands belong to two different people, and then release it, and the subject pinches the ruler as quickly as possible. We can then use some simple formulas, which I'll show and provide links to calculate the reaction time based on how far the ruler dropped before you were able to pinch it. Make sure you do multiple repetitions so that you can average those values together. Then you can repeat that experiment with your eyes closed, or the pincher's eyes closed, and the partner providing a verbal cue or a touch cue, say on the shoulder, at the moment that they drop the ruler, and using those auditory or tactile cues instead of the visual cue of seeing the ruler drop. With two rulers, you can also test how adding this uncertainty and extra processing that needs to happen alters the subject's reaction time. So in analyzing this experiment, you can talk about the different steps that need to happen to transform a visual input into a motor output. First, transferring visual information from the eye to the brain, visual processing to detect that there's a hand holding a ruler based on the pattern of a bunch of pixels, detecting the motion of the ruler that indicates that it's been released, getting that information from visual cortex where it's been detected to motor cortex to trigger and then send a signal to the spine and ultimately cause the muscles to react and pinch the ruler. You can use estimated speeds for action potential traveling down axons from the brain to the spine, say approximately 100 meters per second, and a couple synapses at a millisecond or two each, to calculate how much time neurons are actually processing the signal in the brain. And we do know that, say, in V1, the neurons begin to respond about 40 milliseconds in. So how much time is there between that first visual response in the brain and the motor signal being sent out the spine? How does this change? How does this processing time in the brain change when there are two options, two rulers, instead of just one? Is there any difference for visual versus auditory stimuli? So hopefully this little experiment can get students thinking about the idea that even for very simple reactions, there are a number of processing steps that need to occur in the brain, and that the elapsed time can provide a window onto that processing. And we saw in the psychophysics experiments how a cue that directs attention can speed up that reaction time by facilitating those processes. Another experiment that doesn't require any fancy equipment is to measure tactile acuity on different parts of the body. So a partner is going to poke your skin with either 2 points of pressure or just one, and the 2 points are going to get progressively closer together. And at some point, you'll lose the ability to tell the difference. The subject's eyes are closed, very important. At some point, the subject will lose the ability to tell the difference if it's 2 points of pressure or one. So exactly how close those points will need to be is going to depend on where on your body you perform the test, because some parts of our skin are more sensitive than others. You definitely don't want to use your fingers for this test. Calipers work great. Or you can cut points out on a business card, and I'll give you a website that has a template for cutting out those little points on a business card. You can attach toothpicks to an index card at different distances that you've measured and just tape them on there. And then you can use those toothpicks at different distances to find that threshold separation. So once you've measured that threshold separation at multiple parts of the body, you can enter that information into this homunculus generator software, and what you'll see is a cartoon of the body scaled according to the sensitivity of the different parts of the skin. When you're doing your measurements, you can get much more specific than just the heads and hands that they use in this cartoon. So you can measure differences between lips, cheeks, forehead, scalp, your fingertips versus the back of your hand or the palm of your hand should give you different values. So you can get as specific as you want when you're constructing these measurements. Now, as far as tying this back into more general neuroscience concepts, we talked about receptive fields in the visual system, the part of space that a neuron is sensitive to. Somatosensory neurons, which are going to sense this pressure, also have receptive fields. This is an area of the skin where they're sensitive to a pressure stimulus. And this corresponds to where the dendrites of this neuron are physically located in the skin. Pressure on the skin then opens mechanically gated ion channels in the receptor neuron's membrane, causing an action potential. And the brain knows that when this particular neuron is firing, something must have touched the skin within its receptive field. Given this background information, you could talk to your students about whether neurons with larger or smaller receptive fields would provide higher tactile acuity. Imagine the responses of two neurons with either large or small receptive field to pressure at two different points at different distances apart. How will the neurons respond to one versus 2 points of pressure at different separations? And you can just make these simple on or off responses. Either there's a stimulus in the receptive field or not. Which size of receptive field will allow you to tell points that are closer together apart, based just on the responses of those neurons? If you have smaller receptive fields, will you need more or less neurons to collect signals from the same area of? Then you'll also need more cortical neurons to process this more detailed incoming information, which is exactly what we see in primary somatosensory cortex. More cortical area is devoted to processing signals from areas with greater tactile acuity. This also ties into the idea of neural resources being limited more broadly. So you could have the same sensitivity that you have at your fingertips everywhere on your body, but this would require many more primary sensory neurons and more cortical neurons to process those incoming signals. Just look at that cartoon image of the homunculus you made and imagine how big it would be if everything was expanded to match the scale that the fingertips are at. All those neurons would be using energy to manufacture neurotransmitters, to transport sodium and potassium ions across their membranes to maintain their concentration gradients. This costly aspect of neural processing is a large part of why we need attention to select objects and focus and devote our limited neural resources to processing the most behaviorally relevant information.